Thank you very much to the Forever Healthy Foundation for inviting me to give you an idea about our new company, the Rockfish Bio. Before I do that, I would like to introduce myself. My name is Otto Kanzler. I'm more than 40 years in the pharmaceutical industry. I spent 25 years at a big pharmaceutical company. And in 2011, we founded our first company, a biotech company dealing with the development of tools, biotechnology tools, new tools, following by another company with dealing with biomarkers. And Mehmet, you're absolutely right. Biomarkers are absolutely a very important issue. And our last baby now is Rockfish Bio, where we're dealing with the promoting of healthy aging with senolytic therapies for age-related diseases. So that's the topic. So I guess this is which goes in the next direction. I'll try to do this. So why the name Rockfish? We sit together and discussed, well, we're talking about aging and especially healthy aging. That's absolutely important for us. And then we looked a little bit around, and there are some animals on the planet existing, a fish, a sea-based fish, and it's known that it lives for 205 years at least, but much more important, in good health. So, and I thought, well, we are looking for doing innovative stuff to increase the good health in the life. So that's why we've chosen Rockfish Bio as a name or a brand of our innovation. I don't want to repeat it again because Mehmet mentioned all the stuff around that. We have an aging population. That is good news. The population 65 plus will increase dramatically during next year. And the question is how we currently age. And that's probably the bad news. And Mehmet mentioned this as well, so I don't want to repeat it again. So there is a huge economic and ethical value there for the society to improve that stuff, that we live longer, especially healthier. That's what drives us at the end of the day. So the dream is living in a world without any age-related diseases or disorders. Getting old in good faith. So Mehmet mentioned that as well. I have to mention him very, very often. The dream will partly come true. It's a question of the time. Yeah? And we do believe that we will be a part of the realization of the dream. So one hope in that direction are senolytics. Everybody knows that eliminating of senescent cells will reduce the development and the progression of age-related diseases. So that will be delayed. And there are many, many diseases there which are known in literature, and you know it better than I do. So definitely that's an issue. So if you look a little bit about the current landscape, about the companies who are dealing with senolytics, so we see that the first generation of drugs has some drawbacks, let me say in that way. So they are not effective in all cell types. They have low selectivity and severe side effects. This is mainly based due to the fact that people are using drugs which have originally been developed for cancer. So therefore, we have severe side effects. So therefore, there is a huge unmet need to find new concepts, new ways to eliminate senescent cells selectively. Another issue, which is we have the spoil of choice. Which indication should we go for? So we have analyzed a little bit up to 40 different indications which are related to senescent cells. And if you, for example, have a drug which attacks a specific pathway, so probably it works in one of the 40, probably in another not. So that's definitely an issue. And how can you address this? To find the right indication, probably. So the point is, you need to have really good biomarkers to see what works. Mehmet mentioned this. I'm fully in line with him. So that's very important to have all these issues addressed. So Rockfish, the solution, we have developed a revolutionary target pathway where we do believe that could be the game changer in healthy aging. So the key figure is that we've seen that senescent cells have increased phospholipase A activity in senescent cells. I will tell a little more later about the mechanism. So what we have seen so far, that the intervention in this pathway is effective in all tested cell types and organs, which we tested so far. We are talking about 10 different organs and liquids which we have tested. We have seen in vitro tests that we have really a huge therapeutic window, so that will not happen at the end of the day in the humans, that's for sure. But nevertheless, there is a huge window there. And we have excellent preliminary in vivo data in naturally aged mice. So, and together with our partner company from us, we have developed a biomarker estimation to give you an idea about the load of the senescent cells in the body. And we will use this together. So how does it work? I mentioned that the PLA2 activity is increased in senescent cells. What does PLA2 do typically? Typically, PLA2 is responsible for the production intracellularly of arachidonic acid. So that's a fatty acid which is very important for many pathways in the cells. And that will be converted by 3 main enzymes, COX, ALOX, AXL, and will be transported to the other pathways. In the senescent cells, we see that the concentration of the arachidonic acid, due to the higher production rate, is increased. But the enzymes are powerful enough to convert this out of the cells and progress it further. So then we thought, what about if we partly inhibit the conversion of the COX, ALOX, AXL enzymes? Should that lead to an increased concentration in the cell? And for sure, that's definitely the case. So if we do this by blocking with a small molecule, which is our lead candidate, then we reduce the conversion of arachidonic acid out of the cell in the next pathway. Due to the fact that in senescent cells we have a much higher production rate of arachidonic acid, the concentration in senescent cells reach up to a lethal threshold. Yeah? And due to the high intracellular concentration of arachidonic acid, the cells goes to death, apoptosis. So since we're talking about the intervention, a treatment of maximum 14 days, yeah, after this 40 days, the concentration of arachidonic acid in the healthy cells goes back to a normal level. So that's in principle the concept. Yeah, and we have tested it, and it works in many all tested cell types. As I mentioned, we have to develop together with Tamirna a biomarker showing, give you an idea about the level of the senescent cells. This is very important for development work. That's very important for clinical trials to find out the right patients. So patient stratification is definitely an issue. And later on, it's important as well for the treatment monitoring and for the patient stratification later on to develop, to define which patients should be treated. So that's a very important issue. This test is available, and we are optimizing this further. Since it works in many different cell types and organs, we do believe that we have a multi-indication platform for the treatment and much more for the prevention of age-related diseases. Mehmet mentioned that as well. We need to advise a little bit, to train a little bit the authorities, because currently it's not possible to run preventative trials in a short way. So that's very time-consuming and expensive. So therefore, our strategy for now, yeah, Later we need to do this in a different way, of course. For now is to look whether we find probably a rare disease for a fast-track approval. In addition to that, we will generate preclinical efficacy data in 4 to 5 different additional indications. That's a question of the money. If we have more money, then we can run additional data in 10 to 20 different indications. So it's a question of scaling at the end of the day. And as soon as we have an approval for one of the indications, we will promote the off-label use for any other indications. So that brings me back to slides from Meho, where he said, well, we need to use the media, we need to use the public information, we need to call it, we need to tell them this, all the stuff, the society and the community. So that's which needs to do. That's our strategy there. So that's just a short slide to show you how we ranked the different indications. So we made a score, and for the time being right now, we have identified that probably idiopathic pulmonary fibrosis, which is ranked as a rare disease, should be our main indication. Our vision for the treatment of senolytics is first do an analysis of the senolytic cell test using our Xenomere tests, probably on the routinely health check you do. You see whether you have a high level of senescent cells. And if that's the case, you should start the senolytic therapy to reduce the senescent cell burden. So that will be probably oral delivery for a time of 1 or 2 weeks. Then after the treatment, we do the reanalysis to see whether the treatment was successful or not, and then we do the retesting every 1 to 2 years, and to recycle the treatment if necessary. And additionally, and we need to think a little bit how to do this, we would like to promote the preventive treatment in high-risk population. The mechanism needs to be developed. We are not at that stage right now. Just some brief information, and I don't want to show you all the slides here, or the data. So we have seen that it works in kidney, in liver, in spleen, in fat, in skin. We have seen, and that was very surprising for me, honestly speaking, that our lead candidate using this strategy improves the physical fitness of the mice. On the one side, you see a hanging wire test. Mouse hangs on a wire, and you see we measure the time till it falls down. And mice treated, we're talking about mice with an age of 27 months, which relates approximately to 75 to 80 years of the humans. We are more or less in the same level than the younger mice compared to the not treated mice. And we've seen there's a huge improvement in the muscle function and in the weight. Then we did a frailty index, and you see the difference. Frailty is absolutely exciting, in my opinion. And the open field test, exploratory behavior, shows that the young mice are walking around. The old mice are not that fast moving, and using our truck, you see that there is a more exploratory behavior there. And if you look at the pictures, the one mice has gray hair and less hair, and the other has black hair. So I will take probably the drug in 1 or 2 years, and we will see whether I get hair again. So another issue what we did extend health and lifespan. And you see we had a group of approximately 30 animals for each group, the days of survival after the treatment, and the overall age at death. And you see there is a shift. Please keep in mind that the mice are 27 months old, so 75 years. What about if we start the treatment with 60 years, or, as Mehmet mentioned, 40 years? We have no idea right now, but we see definitely a huge impact. Yeah? And compared with the physical fitness that the mice running around, we can think that probably the overall health is better. We have no correct data so far. Our plans for the next is, of course, we need to look what the drug is doing in the body. We need to optimize the drug. We have no idea right now about PK and ADME. Therefore, we need some money and some funds. And we would like to implement some disease models to get information about the lead indication, IPF, and of course, probably other disease models if we go in different ones. And the optimization of the companion diagnostics anemia. Yeah? So there are some ideas around that what needs to be done, and we would like to go in that direction. So the market potential is huge. Mahmoud mentioned this, so I don't want to repeat this again. So it's different kind of estimation, but it's definitely huge. Yeah. So we are looking currently for a Series A round of approximately 5 million or more. And if we partner with somebody, we have probably not an exit strategy, but we could have an exit strategy after clinical phase 2A for each indication. Alternatively, any other concept from licensing, partnering, and IPO is possible. And we expect the exit for the first indication in 5.5 years. I would like to come back to a comment from Mahmoud again, who mentioned collaboration is a key. I definitely agree with that. We are looking for collaboration partners as well. Yeah? So we are very open to this, yeah? Because we definitely know that even we have Rockfish and even if we have Tamirna in our company portfolio, let me say in that way, that's definitely not enough to be as good as possible and as fast as possible. Yeah? So we're very open to discuss any such collaborations. So the team, Otto is me, Ingo Lemmermann, our CSO, is here with us, and Johannes Grillari is, let me say, the scientific head of the story. And for that, I would like to invite you to become a part of our exciting story, which could be probably one of the largest, this is in the pharmaceutical history, and to help us to achieve a life without age-related diseases. And I'm open for any discussion, so let's talk. And thank you very much for your attention.